protein.
What we now know as prion-related diseases such as scrapie in goats, bovine spongiform encephalopathy or mad cow in cows, and Creutzfeldt-Jakob disease in humans were in the recent past not understood by scientists. As the video you just saw depicted, even in the mid-20th century in the 1960s and 70s, scientists such as Michael Alpers and Daniel Gajduszek, who saw the effects of prion disease in the Kuru populations of New Guinea, didn't yet understand the complexity of how the proteins were transmitted from organism to organism. When Daniel Gajduszek won the Nobel Prize in 1976 for his research in how Kuru was spread from person to person through infected tissue, the structure and mechanism of prion proteins were still unknown in science. In 1997, prion proteins were finally discovered by American biologist Stanley B. Prisner. Prisner described prions as transmissible particles devoid of nucleic acid and composed exclusively of modified protein, PRP-SC. What Prisner was describing was the misfolding of a protein that exists in human brain tissue, PRPC or prion protein. Prion proteins exist in human brains without causing any harm until a malformation of the proteins causes them to misfold into an abnormal conformation, PRP-SC. The misfolded state of prion proteins, which is characteristic of prion disease, has a number of key features. Firstly, a highly conserved network of hydrogen bonds in polar clasps, which contributes to its stability. Also, it is insoluble and multimeric, meaning it has multiple peptide chains. The misfolded prion proteins have a notably higher concentration of beta sheets. Beta sheets and alpha helices being secondary protein folding structures. The significance of having a larger number of beta sheets is that, as mentioned previously, this contributes to the abundance of hydrogen bonds in their structure, leading them to be more strong and rigid than natively folded prion proteins. It is suggested by existing research that the misfolding in prion proteins occurs because of a core principle. Proteins are constantly changing how they are folded and adapting new conformations. The more efficient, meaning the less effort the protein has to use to keep a certain folded formation, the more likely it is to gain and then keep that formation. For prions, the misfolded state is 900 times more efficient than the native healthy state. This means that while normal proteins can refold or unfold and change their conformations, once prion proteins have adopted a misfolded state and begin to aggregate or group together, such as in protein aggregates called amyloids, it is extremely difficult to induce the proteins to any other conformation. The aspect that is so damaging about prion protein misfolding is that it isn't isolated to single misfolded proteins. One theory suggests that once one protein has taken on a misfolded state, it imprints its misfolded conformation on other nearby normal proteins, causing them to misfold as well. This theory is called template-directed refolding. The initial misfolding step occurs sporadically in brain tissue, which happens infrequently and is why genetic prion diseases such as Creutzfeldt-Jakob are so rare. But as well, the consuming of tissue containing misfolded prion proteins is primarily how the misfolded proteins transfer from one organism to another. As the earlier video mentioned, a devastating neurodegenerative prion protein-related illness called Kuru spread rapidly in a human population in New Guinea who are known to practice cannibalistic practices. It suggested that in this population, one initial sporadic case of genetic prion misfolding in the brain was spread throughout the population due to the ritual consumption of brain tissue as part of funeral practices, causing large numbers of people to become sick with the same prion misfolding disease.
So in 2016, it was found that the copper ion induces oligomerization of the prion protein. So what does that mean? Well, it means that on a molecular levels, the copper ions cause the prion proteins to misfold incorrectly, and then these misfolded prions actually stick together 900 times more efficiently than the healthy ones. So this is a diagram showing amyloid, which is what they call this protein mass of a misfolded protein. So on the left here, you can see different proteins. Our prion there is in blue, that then misfold. They call that nucleation and form a clump. And then they grow to fold these amyloid fibrils, and they all have this common amyloid fold as represented on the right there. And here's a nice drawing as well of some normal neuron cells on the left, and on the right in Alzheimer's disease, you can see these amyloid plaques. That's those amyloid fibrils, the clumps of protein. And in this case, for Alzheimer's disease, the protein is not prion, but tau protein, TAU. Another disease is chronic wasting disease, which is now a global pandemic for deer and elk. In this diagram, you can see that the prion protein amyloid fibrils, they clump up and disrupt the neuron activity in the brain. They collect in the eyes as well, the spinal cord, and this can obviously disrupt proper neural transmission of signals and sensory information. And one of the questions here, obviously, that's relevant very much to the public that they're wondering about and worried about is, can chronic wasting disease transfer from elk to people? That's indeed what happened with bovine spongiform encephalopathy, which is commonly known as mad cow disease. And this had sort of an outbreak in the end of the last century, 1980s, 1990s, when there was indeed transmission through food of people eating cow meat. And other diseases include scrapie in sheep and goat, and Creutzfeldt-Jakob disease in humans. So here's an interesting study I want to show you, in which the researchers actually developed human cerebral organoids. So an organoid is like a mini sort of simple model of an organ. So this is the growth you can see from left to right in a petri dish of almost, I guess, a mini brain to model the disease in this human organoid to better understand the progression of the disease and some therapeutic targets. So what sort of targets we can attack.
So while some aspects of prion misfolding have been described in science, there is a large amount of work to be done. The specific relation of prion misfolding to diseases such as Alzheimer's is not well established. Therefore, there are many scientists who are currently trying to work on new methods of treating these diseases, but even more work must be done.